Welcome to our explainer. Today we're going to talk about a single protein, one that holds the key to both protecting our brain and, weirdly enough, causing its complete destruction.
So get this, what if the very protein designed to protect your brain could also be the thing that destroys it? I know, it sounds like science fiction, but it's not. It's the real-life story of a fascinating molecule called Neuroserpin, and we're about to dive deep into it.
Okay, so before we can really get what Neuroserpin does, we have to talk about where it works. And its office, the human brain, well, it's basically under construction 24/7.
I mean, think about it. Every single time you learn something new or make a new memory, your brain is literally rewiring itself. We call this neuronal plasticity, and it's incredible. But just like any huge construction site, all that building and tearing down has to be managed, right? Otherwise, you just have chaos. And that's where our protein of the hour comes in, Neuroserpin. You can really think of it as the site supervisor for that non-stop construction project in your head. And believe me, its job is absolutely critical.
So, to really get this, imagine a delicate dance. You've got this one enzyme, TPA, that's like a molecular sculptor. It's actively carving and reshaping our neural pathways. But you can't just let a sculptor go nuts, right? So that's where Neuroserpin steps in. It's the supervisor that walks up, taps TPA on the shoulder, and basically says, "All right, pal, that's enough carving for today."
And where is this supervision needed most? Well, in the busiest parts of the construction site, of course. Neuroserpin hangs out in the brain's major hubs for things like higher thought, learning, memory, and even emotion. I mean, these are the zones with the most intense, non-stop remodeling. Plasticity is happening there all the time.
You know, what makes Neuroserpin so good at its job is how specific it is. This isn't some clumsy supervisor yelling at everyone. Nope, it's a specialist. It knows exactly which enzymes to target, like our sculptor, TPA, and it forms this tight complex to shut them down. But then it completely ignores other enzymes like thrombin, which has a totally different job with blood clotting. That precision, that's the whole key.
So how does it actually do it? How does it stop the sculptor? Well, on a molecular level, it's not just blocking it. It's something way cooler, and honestly, a little more violent. It's basically a tiny, perfectly engineered mousetrap.
Okay, this mechanism is absolutely brilliant. Check this out. Neuroserpin just kind of dangles a piece of itself out there, this little loop, like bait. The TPA enzyme sees it and can't resist. It comes in for the kill and cuts the loop. But bam, that's the trigger. The cut loop violently snaps back into the main body of the Neuroserpin protein, and it drags the TPA along for the ride. The TPA gets all twisted up, distorted, and totally destroyed. It literally uses the enzyme's own attack to spring the trap. So cool. But here's the twist. That incredible flexibility, the very thing that makes the mousetrap so effective, well, it's also its greatest weakness. And this, my friends, is where our story takes a really dark turn.
Yeah, this quote here nails it. To work like a spring-loaded trap, Neuroserpin has to be in what scientists call a metastable shape. Basically, it's intentionally unstable. It's always full of tension, just waiting to snap shut. But that very tension, that's what makes it so easy for it to mess up and fold the wrong way.
And when it does misfold, it's not just a small mistake, it's catastrophic. It can lead to this rare but absolutely devastating disease called FENIB. It's a form of dementia, and it's caused directly by this one protein going completely rogue.
So, in a healthy person, everything goes according to plan. The protein folds up perfectly, it gets into that stressed, high-energy, ready-to-pounce shape, and it's all set to do its job as the brain's supervisor.
But with certain genetic mutations, the whole process just breaks. The protein never makes it to that final functional mousetrap shape. Instead, that little bait loop we talked about, it gets stuck. It inserts itself into the next Neuroserpin molecule, linking them together like a chain. And this happens again and again and again, forming these long, sticky, toxic chains called polymers. And these polymers just build up inside our neurons until they literally kill the cell.
So what we're left with is this incredible duality, right? It's one protein with two completely different faces. It is the perfect and kind of scary example of how a protein's shape dictates what it does in our bodies, for better or sometimes for much, much worse.
I mean, look at this. You've got these two completely different identities packed into one single molecule. On one side, you have Neuroserpin the protector. It's essential for learning, for memory. It even helps reduce brain damage after a stroke. But then, on the other side, with just one tiny genetic mistake, it becomes Neuroserpin the pathogen, the direct cause of a fatal brain disease.
a hero or a villain. It all just comes down to its shape.
And all of this leaves us with a really exciting question for the future, doesn't it? If we can really understand this razor's edge between the good shape and the bad, toxic shape, could we actually design drugs that could, say, lock neuroserpin into its helpful form? We could boost its protective side to help with things like stroke, while at the same time completely stop it from ever turning into the bad guy that causes dementia. I mean, think about that. Finding the answer? Well, that could change everything.